Hello, good day. I'm Riosahlie Madrid from Aurora State College of Technology, Baler, Aurora Province, Philippines. And today I'm going to present my research entitled Development of Reference Template for Lump Assay in Detecting FGCAT-B3 Gene in Juvenile Fasciola gigantica flukes among large ruminants. In the field of medicine, we have what we call the epidemiological triad wherein there are three factors that can affect the condition of the animal: the host, agent, and the environment. If one of these factors is disrupted, the animal will have a disease or even mortality. And that's one of the factors that the Provincial Veterinary Office of Aurora Province prioritized since 2017. As we can see on the graph, the leading causes of mortality since 2015 is fasciolosis. The province is located at the east-central side of the island of Luzon, wherein rainfall is constant and abundant and is rather evenly distributed throughout the year. This topography is considered for the results of high and low number of mortality in fasciolosis. But as we can see, there is also high prevalence of fasciolosis in other parts of the country. So what is fasciolosis? Fasciolosis is a parasitic disease of ruminants like cattle, carabao, sheep, and goats caused by a parasitic fluke, Fasciola. And what is Fasciola? Fasciola is a fluke of ruminants that is leaf-shaped and targets the liver of the host. There are two species of Fasciola: Fasciola hepatica, the one on the left, and Fasciola gigantica, the one on the right side. The most abundant Fasciola species in the Philippines is the Fasciola gigantica. So how does Fasciola invades its host? When the ruminant defecated infected feces with Fasciola egg, the egg develops into merasidium stimulated by temperature and humidity. This merasidium then penetrates the intermediate host, which is the snail Limnaea auricularis rubigonosa, or the rabid snails in the Philippines, that is abundant in rice field and in wetland areas such as river banks. After penetration, the cercaria sheds from the snail and attaches to the base of the grass. So when the cattle eats the grass with the cercariae, the ruminant is said to be infected with fasciola. So traditionally, through fecalysis under the microscope, we can identify the presence of fasciola in the ruminants. But the presence of egg through fecalysis indicates that the animal is already severely affected with fasciola. This shows the need to develop a reference template for molecular tests that will further validate the possibility of early detection of fasciolosis among animals for early implementation of control or preventive measures. So what is then the newly existed juvenile or marita, and how it can destroy the liver of the host? When the cattle eats the grass, like what I'm talking a while ago, containing the cercariae, it will be called metacercariae. And upon invading the host liver, the metacercaria releases NEJ, or the immature fluke. Within the liver, the NEJ releases catepsins or enzymes, which are involved in the destruction or liquefaction of the liver. It is studied that catepsin B is the major protease from the immature fluke, and catepsin L is released from the adult fasciola. FGCat-B3 gene is an isotype of cathepsin B proteases in Fasciola gigantica, which were cloned from stage-specific cDNA libraries. These findings suggest that this gene may be involved in digesting host connective tissues during the fluke's penetration and migration in the host, making the animal weak and suffers death if not controlled. So there will be an increase in mortality of ruminants, decreased meat supply, shortage of exports, increased demand of meat, increase in price, leading to low income of owner or raisers. So how the reference template was developed. Immature flukes were collected from the liver of dead buffaloes or carabaos or cattle in Aurora province. Marito flukes that measures less than 10 millimeters, according to Dusak, were extracted from the infected liver and were finely macerated for RNA extraction using noctozole with revisions. After extraction, the samples were subjected to RT-lamp assay and were also checked through RT-PCR for confirmation and subjected to gel electrophoresis. Complete genome sequence of cathepsin B3 was obtained from the GenBank to evaluate the primer sequences as shown below. The amplicon size of this gene is 1014 base pair. Figure shows the result of RT-PCR of the Marita. Among the 8 Marita extracted, Marita 7 and Marita 8 exhibited the ladders with an amplicon size of 1014 base pair. In this study, RT-LAMP is more sensitive compared to RT-PCR for detecting the gene cathepsin B3 in juvenile flukes. After that, 2 ml blood were collected from 160 animals from the 4 municipalities of Aurora province. There were 20 cattle and 20 swamp buffaloes enrolled in each municipality, which were randomly selected based on the reported case of fasciolosis from the provincial veterinary office. The animals were distributed according to age groups: 10 cattle or swamp buffalo below 1 year old, and 10 cattle or swamp buffalo above 1 year old. And all samples were immediately subjected to RNA extraction. These tables show the distribution of infected cattle and swamp buffalo that carry the gene that expresses cathepsin B3 in their blood samples according to the methods of providing water, wherein 42% of cattle and 32% of swamp buffaloes that drink water to the rivers or stream were carrying the gene, while 29% cattle and 70% swamp buffaloes that were provided water from the well, stream, or rivers might be infected with the gene carrying cathepsin B3. This figure shows the RT-LAMP assay results of representative blood samples and the multiple ladders from gel electrophoresis indicating the presence of the gene that expresses cathepsin B3 in immature flukes. Table shows that more swamp buffaloes were positive for cathepsin B3 gene as compared to cattle. This implies that more swamp buffaloes may be carrying juvenile fasciola in their circulation because there were similarities and differences in the immune responses. Table shows that out of 44 blood samples, more number of caracoles are positive for the gene of cathepsin B3 than adults. On the other hand, in cattle, more number of adult cows or bulls are positive for the gene of cathepsin B3. Results could imply that young buffaloes were more susceptible to infection than calves and adult buffaloes. It is therefore concluded that repeated detection of fasciolosis in large ruminants can be possible with the use of the optimized RT-LAMP that can amplify the gene expressing the cathepsin B3 enzyme, which is secreted exclusively by juvenile flukes. Blood samples can be examined for patent detection, hence, strategic deworming can be implemented before the flukes reach adult stage and obstruct the bile ducts, leading to death. It is recommended that the laboratory workflow be translated into a kit so that prepatent detection of fasciolosis for strategic deworming can be done. So let's help our farmers or livestock owners increase their profit. Thank you.